Okay, let's go ahead and go over your pedigree worksheet. If you guys have any questions, please feel free to ask them. Let me turn off the light. All right, 1 through 5 are all based on this pedigree. Which members of the family are afflicted with Huntington's disease? That's a disease that basically is neuromuscular degenerative disease. It shows up around your 30s or 40s. So you could theoretically, you could have a child before it starts to take effect. This would be individuals 1, 1, 2, 2, 2, 3, 2, 7, and 3, 3. There are no carriers for Huntington's disease. You either have it or you don't. With this in mind, is Huntington's disease caused by a dominant or a recessive trait? That would be a dominant trait. You can tell because it's not skipping generations. And you often have an affected person likely, what is there? There's about at most a 50% chance that they can affect someone who is unaffected, or they can have a child that's affected. How many children did individuals 1, 1, and 1, 2 have? They had 6. You got 1, 2, 3, 4, 5, 6. If you counted 2, 5, that's 2, 4's husband. That's not their child. How many girls did 2, 1, and 2, 2 have? 2. How many have Huntington's disease? 1. How are individuals 3, 2, and 2, 4 related? Uncle, niece. How about 1, 2, and 3, 5? Grandma, grandson. Are there any questions on 1 through 5? Okay. 6 through 11. The pedigree to the right shows a family's pedigree for hitchhiker's thumb. Is this trait dominant or recessive? Well, we can see it skipped generations, so that's going to be a big indicator that it is recessive. How do we know? Well, 1, it skips generations. And 2, unaffected parents produced affected offspring. How are individuals 3, 1 and 3, 2 related? Cousins. How would you name the 2 individuals that have hitchhiker's thumb? 1, 1, 1, 3. Name the 2 individuals that were carriers for hitchhiker's thumb. 3, 1, 3, 2. Is it possible for individuals 4, 2 to be a carrier, and why? Yes, because if you look at the pedigree, individuals 3, 1, and 3, 2 are unaffected, but they produced affected offspring. Therefore, they're carriers. When you have 2 heterozygotes breed, there's a 50% chance that their offspring will be a heterozygote. If I did the Punnett square, you can see for yourself. Heterozygous, heterozygous, 25% homozygous dominant, 50% heterozygous, 25% homozygous recessive. So why? I would say there's a 50% chance, and I'm going to just abbreviate because I'm strapped for space here, 50% chance 2 heterozygotes produce a heterozygote. So number 2 is likely a heterozygote. There's a 50% chance, and there's a 25% chance that they are homozygous dominant. All right, 12 through 16. Now, if you had a little trouble with this one, it's understandable because we have not done sex-linked traits yet. We're going to do those next week. A sex-linked trait is a trait that is on the sex chromosome X, the X chromosome. It's also called an X-linked trait. The pedigree to the right shows a family's pedigree for colorblindness. Which sex can be carriers of colorblindness and not have it? That would be female. Females, you have 2 X chromosomes. Males, we have 1. And so the ladies can actually be a carrier for a sex-linked trait and not show it. Because the males only have one X chromosome, they either show it or they don't. The other chromosome for a male for sex chromosomes is Y, and Y hardly has anything. So if you look up on the whiteboard, I did that last period. Where's my clicker? Hold on a sec. If that's a female, if you look at dominant B is normal, recessive B is recessive, or little b. If you're a female like this and you have big B, you're dominant, you're unaffected. If you're big B, little b, you're a carrier. You're not affected, but you could pass it on. If you're little b, you're homozygous recessive and you do have the trait. But then look at the males. Males cannot be a carrier for a sex-linked trait. Males either are unaffected or they are affected. They're either dominant or recessive. There is no carrier for males. So if you look at the pedigree, it shows a lot of females that are half circled. You don't see any males that are half circled. That means that males cannot be the carrier. What kind of trait is colorblindness? It is called a sex-linked. You could also call it X-linked. Why does individual 4-7 have colorblindness? Well, she's a female that's colorblind. That's very rare. But if you take a look at her parents, her mother is a carrier and her father is affected. That means that she inherited a recessive X from her mother and a recessive X from her father. So she has a carrier mother and an affected father. Why do all the daughters in Gen 2 carry the colorblind gene? Because father is affected. And all the ladies in this room, you have 2 X chromosomes. Which one did you inherit from your father? His X or his Y? Come on, it's not that hard. What chromosome did you inherit from your father? His X or his Y? You inherited his X. And what if his X has a mutation in it? Well, because you're female, remember, father determines the gender of the offspring. So the father passed on the X to the next generation. This father right here is like that. So all the females in Gen 2, all the daughters, I should say, here and here, their father is colorblind. So those girls are carriers. Does anyone in here have anyone in your family that is colorblind? Who? So because that's your grandfather on your mom's side, is your mom is a carrier. We know that for a fact. There's a 50-50 chance that you would have been colorblind. You tell me that you're kind of colorblind, right? So you got the recessive trait. Okay. Name 2 4th generation colorblind males. That'd be 4-1 and 4-5. Any questions on the sex-linked pedigree? Okay, let's go down here. Try to identify the genotypes of the following individuals using the pedigree above. Individual 3-3. Well, they're shaded in, so they're definitely recessive. Well, how do I know this is a recessive trait? 2 things, guys. It's skip to generation, and you see in generation 2, right here, 2 unaffected individuals produced affected offspring. That tells you that the trait's recessive. Individual 2-1, right there. Well, they are unaffected, so I know that they have a dominant allele. But look at 2-1's father, 1-1. He is homozygous recessive. The only allele that he can pass on to his children is the recessive allele. So individual 2-1 is heterozygous. You do not have to put a question mark. You can actually deduce that. Individual 1-1 is shaded in. We've determined this is a recessive trait, so they're homozygous recessive. Then we have individual 2-4 right here. She is unaffected, so she has a dominant allele, but she produced affected offspring, so she must be a carrier. Is this trait dominant or recessive? It is recessive. How do I know? Explain your answer. It's skip to generation. and unaffected parents produced affected offspring. How can you know for sure that individuals 2-3 and 2-4 are heterozygous? Because they produced affected offspring. That means that they must be carriers for the recessive trait. And then number 3, brown eyes are dominant to blue eyes. So somewhere over here, let's just put dominant allele, brown, recessive allele, blue. A brown-eyed woman, so, oops. I don't know why that just shifted like that. Okay, we have a brown-eyed woman, so circle. Marries a brown-eyed man. Wait, let's go back. A brown-eyed woman whose father has blue eyes, so he's definitely homozygous recessive, and a mother who has brown eyes, so she has at least one dominant allele, and so does the original woman. This woman marries a man who has brown eyes, and his parents are both brown-eyed. So we know that they have at least one dominant allele. They have a son who is blue-eyed. Please draw a pedigree showing all 4 grandparents, the 2 parents, and the son. Indicate which individuals you are certain of their genotype and where there are more than one possibility. So we got Gen 1, Gen 2, and Gen 3. Well, we have the 2 parents in Gen 2 that produced a homozygous recessive offspring. So what must the genotypes of both people in Gen 2 be? They're both heterozygous. Take a look at Gen 1. We have a homozygous recessive father. We have a heterozygous daughter. Do we know, based on the data that we have at hand, for 100% certainty what the mother on the left is for Gen 1? We don't know for sure, so a question mark would be permissible. Now, take a look at the other couple from Gen 1. They produced a heterozygous daughter. Could both of them be homozygous dominant? They couldn't be, because if they were both homozygous dominant, then where did their daughter get the recessive allele from? So here's what I'm going to tell you. You know for sure that one of the parents over here is heterozygous. Which one? I don't know. So if you just put one of the parents as heterozygous, I'm going to say the father. It doesn't matter if you did the mother too. That's okay. The other parent is either homozygous dominant or heterozygous, but we don't know 100%. So we're going to just put a question mark. Any questions? Okay. If you can put that away, let's get into the main lesson for today. Whoops. All right. Okay, tomorrow you have your Latin quiz number 6. You sure? You sure you study for it? I don't know why it's not letting me click this. Hold on a second, everybody. All right. Okay, today we are going to be doing incomplete dominance. We're going to do codominance. And we're going to do multiple alleles. This means that there's going to be more than just your basic two alleles, dominant and recessive. There's going to be more than that. Okay, so when we talked about Mendelian genetics, Mendelian genetics is the notion that one trait, typically a dominant trait, masks a recessive trait. So if you were homozygous dominant, if we were talking about a dominant allele means a horn and a recessive allele means no horn. If you were homozygous dominant, you're going to have a horn. If you're homozygous recessive, you're not going to have a horn. If we're going according to Mendelian genetics, what would your phenotype be if you were heterozygous? Would you have a horn or would you have no horn? You would have a horn. This concept is known as complete dominance. Complete dominance simply means that one trait is masked by another. Generally, the recessive trait is masked by the dominant trait. Well, there is such thing as non-Mendelian genetics. Non-Mendelian genetics says that rather than a dominant trait mask a recessive trait, the traits form an intermediate. So the people that came up with the whole notion of the blending concept of inheritance, they were thinking, ha ha, told you so, up to a point. So one example we can use to prove this, actually before I get to that, this is known as incomplete dominance. It's also known as incomplete penetrance. Incomplete dominance is going to basically be black crossed with white results in gray. It's an intermediate. It's halfway. And a classic example of this is going to be with flowers. So in some species of flowers, they're called snapdragons, you have two different types of alleles. You have the R allele, which is going to represent red color, and you have the white allele, or the W allele, which is going to be white. So this genotype is red, this genotype is white. What about RW? What do you think? Dan, what color do you think would form from RW? Pink would be it. And so this goes back to the people that came up with the blending concept of inheritance. If you have a red flower, you cross it with a white flower, you would expect to get 100% pink. And so as I said, they're thinking, haha, told you so, told you so, told you that the parents are going to be, or the offspring is going to be an intermediate of the parents. All right. Cross two pink ones then. So what I would like you all to do at your table is I want you to make a Punnett square crossing two pink flowers. And I want you to tell me the percentages of the phenotypes and the percentages of the genotypes. They should be the same. So go ahead and do that at your desk real quick. It should take you just a moment. All right. So you have pink, cross with pink. 25%, what would you, that's 25% red. Anybody want to take a swing at what you would call the genotype for this? Can't say homozygous dominant. Ayla, you want to try? What would you call the genotype for this? There's two different possibilities. Yeah. No, we don't say letters. We say names. Like if I were to say homozygous dominant, heterozygous, homozygous recessive. Okay, true breeding red would be an acceptable answer. Another one would be homozygous red. What would you call this one, Catherine?
True breeding white.
What's another name, Duncan? Yep, true breeding white or homozygous white. What would you call these two, Reagan?
Heterogeneous pink.
You would say heterozygous
Heterozygous pink.
Heterozygous pink. That would really be the only one. Those two are hybrids. They're going to be mixtures of the two. So here's the problem. The problem for the people that were bragging about the blending concept of inheritance. How come when I mix pink and pink, I get red and white? They didn't really have an answer for that. So this just goes to show that with some traits, with some species, one is not going to be dominant to another. They're actually going to form an intermediate. Another example, guys, is sickle cell anemia. Sickle cell anemia is a mutation that originated in Africa. What happened is that RBCs, red blood cells, have the protein hemoglobin. Hemoglobin allows RBCs to carry O2. So hemoglobin is a very important protein for your blood. And when I say protein, what are the monomers of a protein? Unit 1 question. Very good. They're made of amino acids. Well, the way that sickle cell anemia happened is that one of the amino acids was mutated, and it changed to be a different amino acid. Imagine this. Check this out. Let's say that normal hemoglobin, you guys aren't here yet. We're going to learn about this more in Unit 6 after Christmas break. Normal hemoglobin, let's say should be A, B, C, D, E, F, okay? But let's say the mutation is A, B, C, Z, E, F. One letter difference is what mutated the whole protein, and what it does is it causes a normal red blood cell to mutate into a sickled red blood cell. And anybody that has this mutation can trace their ancestry back to this little corner of Africa where this mutation originated from. Over my career, I've met several students who said that they are carriers for this trait. They could trace their ancestry right back to this small little corner of Africa. Now, here's the point I'm getting to. If an individual is homozygous dominant, they do not have this mutation. I would assume that all of you are homozygous dominant. So in this case, you are unaffected. Now, if you're living in Africa, this is actually going to be a problem because do you know what the most deadly animal in Africa is? Mosquitoes. You think, well, they have lions, they have leopards, they have hyenas and crocodiles and black mamba, snakes and giant spiders and all this crazy stuff. A mosquito? Because what do mosquitoes do? They spread diseases, HIV, they spread malaria, they spread dengue fever, they spread yellow fever. And I want to focus on malaria. Malaria is a parasite that is spread by mosquitoes. When a mosquito sticks its proboscis into an animal that's parasitizing, it drinks up the blood, and when it does that, the parasite goes into the bloodstream of the animal. The parasite targets the red blood cells, and it goes inside the red blood cells, and it reproduces within the red blood cells, and eventually the red blood cell gets filled up with malaria parasites, and the red blood cell explodes. And so all the little baby malaria parasites are released, and they all go to infect other red blood cells to repeat the life cycle. This causes anemia. Anemia is a low red blood cell count. You need red blood cells to carry oxygen. So if you have low red blood cell counts, you're anemic. You might also be hypoxic. Hypoxic means low oxygen. You see hypoxic, low oxygen. Well, if you are unaffected with this mutation, the good news is you don't have to worry about sickle cell. The bad news is you do have to worry about malaria. Now, if you're homozygous recessive, you are affected. This means that your cells are going to turn into that sickled shape. And here's the downside of that. These blood cells can be like a big logjam. If you've ever cut your finger and you had just a little bitty incy-wincy drop of blood, believe it or not, you actually cut microscopic blood vessels. We tend not to believe things we can't see, but these blood vessels are so small, you can't see them. And because they're so small, red blood cells have to travel single file to get through. Well, if you have a bunch of sickled blood cells clogging up the works, that could cause a blood clot. And if this blood clot happens in your lungs or your heart, you could die. It's called a pulmonary embolism. So the good news is if you have sickle cell anemia, malaria can't infect that. The bad news is you can get blood clots. But then here is the intermediate. If you are heterozygous, you are going to have sickle cell trait. This is an example of incomplete dominance. This means that you do have the trait, but it's not going to be as bad as if you were homozygous recessive. So you may have some resistance to clotting, and you may have some resistance to malaria. So you're kind of getting the best of both. This is known as heterozygote advantage. You know, what if there's a species of snake? If you're homozygous dominant, you're blue. If you're homozygous recessive, you're brown. If you're heterozygous, you're green. And what if this snake has an enemy, a predator, that can't see green? It can see blue, it can see brown, can't see green. That would be heterozygote advantage. So the hybrids, in this case, a hybrid wins out. It's a benefit. Now, remember that codominance, or excuse me, incomplete dominance is like black and white makes gray. That's gonna, hopefully that analogy helps you out, guys, because codominant is easy to confuse with in complete dominance. So let's get to codominance now. In codominance, black crossed with white makes black and white. There is no intermediate. Both traits are expressed equally. There's no intermediate, there's no halfway. You're going to have both. So if eye color in humans was codominant, your mom had blue, your dad had brown, you'd be blue and brown. Like both eyes would be a little bit of blue and a little bit of brown. Kind of like a tie-dye. So the best example with this are the ABO blood groups. Not only are ABO blood groups an example of codominance, but they are also an example of multiple alleles. So I'm going to make sure I include this. This is a great example of codominance and multiple alleles. Multiple alleles just means more than a dominant allele and a recessive allele. So it's not just big A, little a. It's going to be maybe big A, big B, big C, little c, big D, little e, all of them, all at one time. So we know that there's 3 alleles in humans. There's the A allele, the B allele, and the O allele. For humans, if you have the A allele, it looks like this. B and C. Oh, I got a little ahead of myself. O. Those are the 3 alleles. You inherit one from each parent. So I want us to do all the different combinations. So guys, this is all multiple alleles. Okay, if you inherit a dominant allele from mom, a dominant allele from dad, that is going to be type A blood. We call this homozygous A, or you could call it true breeding A. I guess you could also call it purebred A. Jasmine, what would this be called? This would be called heterozygous A. So what you guys can gather here is that A is dominant to O. If you have an A allele versus an O allele, the A allele wins. All right? Let me actually lower that. Okay. Let's go to the next one. If you receive a dominant B allele for both parents, Turner, what would you call this? You receive a dominant B allele from both parents. You could call it homozygous B. What else could you call it? That's right. This is going to be type B. I think this is what I am. When you guys give blood, they send you a little card in the mail, tell you what your blood type is. They also do like a free HIV check, stuff like that. Sydney, what would this be? The one I just wrote. Wrong class. Samantha, I meant to say.
Heterozygous B.
This would be heterozygous B. This is type B. Okay? Let's keep it going. What would that be called, Sage? We don't have I-type blood. Look at the key I wrote. Homozygous O. You could also call it true breeding O. True breeding O. Okay, and then we have one more, don't we? What would this be known as, John? No. It is heterozygous, but not A. No. Jasmine? Heterozygous AB. A and B are both dominant alleles. They're both fully expressed. This blood type is the example for codominance. This is type AB, the rarest blood type on earth. This one, everybody, that is codominant. You are going to have the fully expressed A blood type and the fully expressed B blood type. So let's do this. Let's do a little example. We have about 8 minutes. We have time. There's a husband and a wife that are having a little baby. And the husband is type B. So let's just say lover boy is type B. And his wife is type A. They have a baby. The baby's type O, and the husband gets all upset. You cheated on me. You broke my heart. You ruined our family. Is it possible for an A and a B parents to have an O baby? I want you to make me the Punnett square that proves that this is right, that proves that this is possible.
Does the wife go on top or is it
It doesn't matter. It doesn't matter which parent you put where, it'll all work out. Okay, so Sophia, what is the genotype of the father? We'll put the father on top. What must his genotype be?
B and O.
No, say the words.
I, U.
No, you want to say heterozygous B.
Heterozygous.
What would the genotype of the mother be?
Heterozygous A.
Correct. So if you check out the Punnett square, guys, there's a 25% chance that they would have an AB child, a 25% chance to have an A child, a 25% chance they would have a type B child, and a 25% chance they would have an O child. There it is. It is possible. So she didn't do anything wrong. Now, I want to talk to you a little bit. Let's get away from genetics, and let me just teach you a little bit about this whole blood typing stuff. Why do we call it A, B, O? What does all that mean? So there's these things called antigens. Antigens are basically self-indicators. They tell your immune system, I belong here. So if you go to the blood bank and you donate your blood and they tell you that you're type A, that means that you have the A antigen. So for example, type A means you have the A antigen. So all of your cells in your body are labeled with this antigen to basically tell your immune system, don't hurt me, I belong here. Well, also in our body, we have these things called antibodies. This is from your immune system. Antibodies target foreign antigens. Things that should not be in your body. Bacteria, viruses, worms, stuff like that. So, let's go through the gambit here. If you are type A, what antigen do you have? You have A antigens. What are your antibodies looking for? Do they want to target A? No, because if they target A, they're going to hurt you. They want to target anything that's not A. So we're going to say non-A antigens. Everything that, all of your cells look like this. They have the little A markers. Okay, if you do type B, what antigen do you have? You have the B antigen. So what are your antibodies looking for? Anything but B. Non-B antigens. So what antigens are surrounding your cells? B. That's correct. Okay, let's see if you can figure this one out. What if you have type AB? The codominant blood type. What are your antigens? Your antigens are A and B. Reagan? Pass when you walk out. So if you have the A and B antigens, what are your antibodies looking for? Non-A and non-B. Your cells look like this. You have both. And then lastly, fun fact, guys, type O is actually not the letter O, it's a 0. Guess what antigen you have? None. There are none. So what are your antibodies looking for? They're looking for everything. You have no antigen. So anything that does have an antigen, your immune system's looking for. So this is what your blood cells look like. Your whole cells, not just your blood cells. So let's see who the universal donor is and recipient. So we have donor and we have recipients. We have A, B, A, B, O, A, B, A, B, O. Let's get this straight. If you are type A blood, who can you donate to? You can donate to A. Who else can you donate to that won't reject you? That's right. If you have type B blood, who can you donate to? B. Who else? AB, because they recognize B antigens as well. If you are type AB, who can you donate to? Just AB. Now, if you think, why can't AB donate to A? Because if you are a recipient of A, B, then you're only A, your body is going to reject the B part. It's going to see the B and go, no, I don't like that. And so lastly, who can O donate to? All of them. So the universal donor is O. The universal recipient is A, B, AB. And believe it or not, despite the fact that O is recessive, it is the most prevalent and common blood type on Earth. So just because something is recessive doesn't mean it's rare. Yeah. RH factor. If you have the RH factor, you're positive. If you don't, you're negative. All right, tomorrow, we're going to start out class with the Latin quiz. Be ready.
I'm not going to do it tomorrow. Shall I put it in or what?
Yeah.
What is RH known?